so happy to see you again, even it's on the screen, and say hi to John as well. I was actually get up at 4 a.m. to listen to John's talk. And so we actually been locked down for almost a year, since January, and all my students cannot come back to the university and to the lab until last week actually. And so we basically, all the projects are stopped, and only projects ongoing is COVID-19 projects. So it's so bad that we don't have a lot of progress on the single cell proteomics that I promised Nicola. Say next meeting I'm gonna to present my result on the embryo development. But still we are working on that, and the paper may coming out in a month or two or something. But for the technology part, I most of the content is the same as last year, but I still have a little bit optimization on that GOAD chip. So I just want to go through the whole journey of single cell proteomics from my side. So why I start to do this single cell proteomics? It's originally from my collaborator in CIPS in Shanghai. Bunch of developmental biologists, they required for that. And one of Professor Lee came to me, give me a single mouse oocyte, say, okay, can you test the proteome for me? And then so, you know, we always proud of our proteomic technique. We say protein ID is easy piece of cake for us. Then so we basically get nothing, and obviously it's too small. So then I think the conventional technique is not fit for the single cell proteomic, obviously. So then the second part is a bunch of clinical people come to me, say they wanted to do a lot of tissue laser capture and microdissection and then liquid biopsy. So those are also very small amount of sample. And then, of course, there is an inevitable trend of scientific development. When you get through all the DNA and RNA, then you definitely will go down to proteome. And so that's the reason for me to basically start this project. So where to start? Then we need to think about, people always say that single cell proteomics is the holy grail. I'm so happy that we have this society built up for 2 years and to start to, you know, solve this very difficult problem. And because you can see this table is from the review in 2008, they list all the different type of cells. So the human somatic cell, it's about 100 picogram level. And then the E cell have 10 times less, smaller. And also the immune cell also very, very small. It's about 10 times smaller. So then we usually, when we do the conventional proteomics, we never deal with picogram level things. Usually microgram, now it's nanogram. So then looking for this, the whole shotgun proteomic process from the first beginning for the cell lysis all the way down to data analysis, you will see too many places that will grab your peptides and proteins to make the loss, the sample loss. So basically, if you want to get a very good, complete single cell proteome, you have to remove every obstacle along the way to get the result. So we decided to start from the very beginning, the sample preparation. So I will think about how to deal with this sample preparation part. So then I think of when you carve, do the carving, and then you use this type of a hammer and a big thing. But if you want to do something on a little rice, so this is basically a rice, definitely the scale is totally different. So that means at the beginning, I thought I need a right tool for that to do the sample preparation, because cell, there's only one little, can't even see the material. and it's too small, so we need the right tool. So basically one goal is to reduce the sample loss and then keep all the treatment process. We don't want to omit any of them, for example, the lysis or the alkylation, digestion. So we don't want to skip any of those process, but we don't want to lose sample. So basically we have to go down to the picoliter droplet, which is the best for reducing surface area. Then start from 2013, I moved to Shanghai from Scripps, and then I was looking for who can do these little picoliter droplets. So then one of my colleagues in the CPMS, she said there was a very famous professor from Zhejiang University, Qun Fang, is a very, everyone knows that it's an expertise of microfluidic. So then I went to Zhejiang University, and then I checked on his publication. So he has a very good method called sequential operation droplet array. It's called SODA. So then the SODA technique is basically a sample, a small nanoscale, picogram scale sample handling. It's like a little reactor. So then they use a very, very fine special syringe pump and then tap the capillary and dip the little droplet onto those well. And so they show me this, they can really go down to 60 picoliter. And so Professor Fang has several patents on this SODA method already. For example, 2017, he has a patent together with Ying Zhu, and also 2016, he has the SODA patent with Li. Later I will mention this girl. And then so have two papers published in 2013 and 2015 about this method. They do this little well on the sample to do those RNA treatment or this type of thing. So that after I saw this SODA, I was very excited, and I proposed an idea to him, say, okay, let's get together. So I want apply this SODA onto my proteomics platform for the single cell proteomic. That was back to October 2013, many years ago. So then he's exciting too, because they do the sample preparation, but they never really done any proteomics in their own lab. They have a very old LCQ classic LCQ DECA, I think, machine. So then Professor Fang sent his student Ziyi to put into my lab in Shanghai to start this project. And basically, we wanted to combine the two techniques for single cell proteome analysis, the droplet-based microfluidic technique and then our shotgun proteomic. The challenge is we have a multiple step sample pretreatment, and also we need to think about how to inject this thing into the LC column. But that's where we start. And then this is basically our analytical chemistry paper in 2018. It's we developed this nanoliter scale oil-air droplets called OTRIP. And so basically it's a little reactor, stationary microreactor. We have a different layer. This is a low-kidney layer, and then the droplet layer, and then isolation, and there's an oil layer. So from this is a diagram show the cross-section part. You can see where the little cell will be in this droplet, and this is the capillary probe, and then they will use the oil to seal this area to prevent the evaporation. And then so you can repeat it to add a different reagent into this little ball. And so the advantage of this GOAD chip is it's easy. It's really easy to handle the picoliter, and also I believe they have been able to perform multiple steps without any complicated handling, and also the oil won't actually contaminate this sample. And so we do this. This is show how we conduct the whole sample treatment process from the lysis buffer and then TCEP and add IAA and the trypsin. And then, of course, we shorten this time, not do the overnight. At the beginning, we did that, the conventional process, and then we inject the sample into our column, just like we did a traditional bomb loading sample loading on the column. So the sample pretreatment and the injection usually about 5 nanoliter to 200 nanoliter. And because we don't want it to see from the first step all the way down here, there's no any steps that you will transfer the liquid or you will do any move this droplet, or there's supposed no sample loss. And so we also create a device, it's called self-assembling monolithic device called SAM, for we particularly load that little droplet, use this high pressure bomb onto the column to do the LCMS. So then we think, okay, that the old chip is really looks like the right tools for single cell proteomics, because we see this is our beginning, give us a single mouse oocyte. Then the first try, we already can get more than around 300 protein out. So we done Ziyi, Dr. Li, she have done many substantially optimization job to make this device be the best performance. So we at the beginning we tried we test the droplet evaporation velocity and to see which material will actually keep this little thing not getting dry. And then so this is what we test, and finally we found the glass looks like very good. And also we tried to see how quick that the droplet can be loaded, so the efficiency of droplet loading. And we found that from keep to the 200 nanoliter, the efficiency will more than 98%. And also we tried different, the material of the sample handling of that chip. And we tried this many different things, Teflon glass and wafer glass and other coating glasses. And then at last we found this PP, this type of hydrophobic material. It's really good. It's actually our centrifuge tube, that type of material. And also because the cell is so small, that means the material is so little, then we thought it may affect our usual for those digestion. So what's the ratio of the protease to the substrate, that we also test about that. Basically, we optimize at 1:20 the enzyme to protein ratio. And then of course, we would do a lot of LC gradient optimization, and then we finally, for this paper, we fix at the lysis, we use RapidJet, and then with the column, we use 20 micrometer, 15 centimeter long. So then back to 2016, the analytical paper, we yet cannot go down to the 1 HeLa cell. So we use the 100 HeLa cell to do the comparison and all the performance. And then so we found that definitely compared to the conventional method, in chip, use this old chip for 100 HeLa cells, we can identify 1,595. And we investigate the sequence coverage, then the sequence coverage actually increased. For example, this diagram shows that use this old chip method, the sequence coverage more than 10, less than 10%, the tube has more, more sample protein IDs, but after that, all the chip is have an advantage about that. Also checking on the gravity value, we identify kind of favor the hydrophobic proteins using this method. And we also find a very strange phenomena that we constantly find some protein show up only in this the chip or the single cell proteome. We don't know why yet, because I told about that last year in Vienna. I said I'm going to detailedly investigate this, but so I don't have these chances since this COVID thing. But we will get back to that. And so after this condition being optimized, so we did try this single mouse oocyte to see what protein we can get. And the amount of that thing, so this is the mouse oocyte size, and this is a single HeLa. And then it's about 20 nanogram. So one HeLa, it's about 100 to 200 picogram. Then this is 20 nanogram. So we get a bunch of oocyte-specific proteins, and also the early embryonic development, and then embryogenesis-related protein, and then very important, the sperm binding protein CP123, and another PABPC1. This is kind of essential for fertility, and also because it's a favorite hydrophobic protein we have, even though from that little amount of ID, we still find the transmembrane proteins and the transcription factor and regulator. So those are quite interesting. For this project, we published in NChem, we actually use a very low-end, well, back then it's a very high-end mass spec back to 2014, and it's an Orbitrap Elite. And then we did that on the fusion, so we later on we have a new fusion. Then we found that the fusion increased the number compared to the Orbitrap Elite. And so after the paper out, then Ziyi get graduated. So then I have another student, so she's working on, well, actually Ziyi continue working on this. We try to improve this oil-air droplet, and this is just a transient version. It's called GOAD 2.0. We basically think it still evaporates because the little droplet still dried up after a while, and then we just make a cover. But now soon we will move this GOAD to 3.0. So we are working on this right now. So this device, we use a completely new material to build this chip. And then this material is corrosion high and low temperature resistance and reusable. So why we want to build a new material? Because we, during the past device, we found the lysis process not really satisfy me, because we cannot really, even RapidJet is the best already, but I still wanted to lysis the cell better. So, and then for this part, we use the liquid nitrogen, and so this device will have to be able to handle the very low temperature. And also that will make the lysis more efficient. And also use these guys, there is absolutely no evaporation. And because we also want to make it like you can totally, fully automate it. And so now we working on that project is use the single human oocyte. So you can see this is a two little picture from this device. And also for this little thing, we have particularly make a flow slot, so you can externally connect to many different things. For example, the circulating water bath and the liquid nitrogen pump, and you can heat and you can do many different things. So through this this design. And this picture just show you of all the evaporation tests. So from the beginning and all the way down to 1 hour and 20, actually it can hold about 3 hours. We're just not taking that many pictures for this little droplet. And also because it's you can assemble this thing into a fully automated stage, can do self-sorting and picking, and also can connect to different device. And the droplet for our new design, it's really small. We can handle up to 10 to 100 picoliter. And but another part is right now we were using, that's what we create we built a multiple head high pressure loading vessel. So altogether you can load 5 or at the same time, and then we have a bunch of them tandem, so you can have many of the little cells work on together. And it's very efficient, but just it's half automated. I don't want to say it's a fully automated. You still have to manually change this column. And so then to this stage, we then move to the second and the third and the fourth pass. They all need to do more optimization job for that. So another issue is column sensitivity. We try a bunch of different columns, and later on we think that the 20 and 30 micron ID together with the 1.9 micrometer particle and with the 25 length do 2 hours run or 3 hours, it doesn't really matter, and give you the best result compared to the other combination. I'm still thinking, is the plot column a better option? Because I was fascinated by Barry's paper, I think it's in 2015, and assisted with the acoustic lysis technique. So they are able to already get more than closer to 2,000 protein ID in 50 cells. And so, but so they will use the plot column in that paper, but I never know how to successfully pad a plot column. And then I tried to find some collaborator and they all got failed for some reason. So I'm still, this is, I really wanted to try. And so, of course, the mass spec, you need a powerful one. And lately, when I moved to Beijing, I'm able to get this HFX. So to see this mass spec performance in my lab, I'm able to make these guys with a 2 microgram injection, and then I can get 6,000 and up to, this is the most I have ever got, is a 6,888 ID. And also I have several timsTOF probes in my lab as well. And I think they are very equivalent to Eclipse and Exploris and 480. And so for this toy, we are constantly for the HeLa cell, and we do 200 nanogram is a 0.2 microgram. We constantly get a 7,000, more than 7,000 protein ID. So you can see the powerful mass specs really change the game. And so, but when we use the GOAD 2.0, so we still use the DDA with the HFX. This is the condition that we optimize. And then we go down to, now we change this, we're not using HeLa because we don't find, so HeLa sometimes they have a quality issue. They kind of fluctuate quite a lot. And then due to our another project, it's a precision medicine project with the lung cancer, so we actually we just changed to A549 cell line. That's a small cell lung cell line, lung cancer cell. So for the one A549, and then this is our newly result used HFX, and one cell like 200 picogram, we're able to get 923 match between run. So we basically always use the peaks for the database search now. And particularly quick on this figure is Sunny was mentioned about the copy number per cell that he's interested in to look, and this is what we get from the A549 cell. So we actually identified the lowest was 108, and the highest we are able to get a 1E8. So that is the region. And so let's see about the SCP current ID comparison. And this is the Barry's paper that I just mentioned. And so constantly, I think it's quite similar. When you use this the small sample treatment, and then right now for the HeLa cell, one cell about 200 picogram yesterday Brian Kelly already presented, they can identify about 800 to 700 to 800 protein. And then for us, for the A549, we did measure that's about 200 picogram. Using the HFX, we can do similar region. It's from 600 to 923 proteome. And for the peaks, for timsPro machine, and use the peaks, particularly you have to add the CCS value together with that. We did get a higher number. It's from 970 to 1,360. That's the highest number I ever get. I haven't over this number yet. I think it's quite similar to yesterday Alexander's talk when they used the DIA together with microfluidic. I think the level should be there. It is quite true. So which means when you use the standard peptide, you will have a little bit higher, and then when you use real cell one, then you can get a little lower end, but that will be different, about 100 or 2. And so for me, particularly tracing the number is not the main goal. We actually just want to be qualified for the game. So we wanted to use this method to apply into the project. Remember at the beginning, I say that basically I started a single cell proteomic project is because I have to meet the requirement of the developmental biologists first. So then our famous Jie Chao from Peking University Third Hospital. So she collaborate with Fuchou from Biopeace. Sunny also mentioned yesterday, the woman who made the Mabel baby, together with Sunny. And I said they already very famous in the field of single cell everything, RNA-seq and DNA sequencing. So this is the 2 years ago, they have a Nature Structural and Molecular Biology paper, and they use a single cell RNA-seq profile the human pre-implantation embryo and embryonic stem cell. So then they want to see, so after you get all the gene things, you definitely want to see the function. So they wanted to know what the proteome of this cell. And so these are, in that paper, they use this different stage. The embryo were at 7 crucial stages of pre-implantation development. This is from oocyte, and then the zygote, and also 2-cell embryo, 4-cell. So the cells start to divide it and into different number. And this is the A-cell embryo and the morula and blastocyst. And so this 7th stage, you can see the size of the cell, they getting smaller and smaller, then they differentiate from that all the way down to the morula already compared to that big wood cell. There's almost like more than 10 times with the size. And so they use this each cell to do the single cell seq. And then so we done the same sample with different job to find out what the proteome of those each cells from different stage. So then we use the same set of a sample. Of course, they're from different donor, but the same stage of this embryos. And then so this is the one oocyte, and this is well, this is a HeLa, but that's almost close to the blastocytes. So then this shows the size per single cell of this embryo at the 7 different stage. And the G1 is obviously it's the biggest one. It's about 125 micrometer. And so we are able to identify about 3,000 proteins. And then all the way down to zygote, it's about 100 similar, but well, sometimes we filtrate. So this is the part. And then we have a 2-cell. Then after that, the cells start to split, and with 2, and the size will go down. And for some reason, we did a better job on that. But then you can see when the cell size is about 65 or above, we can averagely identify about 3,000 proteome. And then after that, the cell getting smaller from down to 40 micrometer all the way down to blastocyst, it's about 70. So HeLa, it's around 15 micrometer. So then we will be able to stick at 1,500 as the average protein ID. So it's making a lot of sense. And well, you can see, you know, when you do the single cell proteomic, for me, I'm quite sensitive of what the size. Every time when the collaborator comes say, can you do the single cell for me? Then I will say, hey, how big that cell is. So I usually will have to very sensitive to what I'm handling. And so then this is the cluster, because I cannot present more data because the manuscript we are under preparation. I just want to show that the quality of our proteome data is very good. And so this is in the NSB, the clustering of their transcriptome of single plasmid with this stage. And this is our proteome of the single plasmid on those from GV. Well, we didn't count GV, but we also did that from oocyte all the way down to the blastocyst. And so that's basically my work to report here. And then the ongoing future is I have a lot of work to do. First of all, I will start from last year. I already wanted to do the DIA, and also I want to put a GOAD chip together with scope 2 for the better quantitation. Right now we were using the label-free corn. We are already working on actually the embryo thing we indeed using the DIA. DIA method. So we also wanted to fully optimize it and make it more robust and make it cheaper. And so constantly or routinely identify about 1,500 protein from a cell. I think it's my near goal. And then I have some other applications, particularly related to some cancer research. And then another big goal is I always want to ask why can we and how to get 5,000 protein or more. Well, 5,000 should be a target first out of one cell. So that may need a really big breakthrough from where maybe it's from the data handling or the mass spec part, because we still have a lot of low abundant molecules buried in that grass, so we need to pick them up. So maybe that's another hope for it. So then that's all for my talk, and I also like to thank my collaborator, Professor Fang and Ziyi and also Ming. Without their help, and of course this project not gonna work. And also now Zhu Liu, so she's also from Professor Fang's lab. Now she's my PhD student. This is my group. This is my the Center for Precision Medicine Multi-Omics, and this is the lab. So thanks for Nikolai give me some time to clarify some of the attribution of the credit and get some justice back. Maybe some people, audience will ask why the GOAD chip and the nanopores are so similar. Well, yeah, I would like to, I was supposed not, I didn't plan to tell about this in this meeting because last year in Vienna I already told some of the truth, but because yesterday I saw someone's name has been brought as a hero, then so I think I should get some justice back. No, it is not because of the great minds think alike things, it's an indecent behavior. So Yinzhu, so he's actually from my collaborator, Professor Fang's lab in Zhejiang University, who developed the solder. They together have several patents and 2 papers. And then, but the nanopores, it was my original idea, in fact. I proposed this idea to combine solder together with shotgun proteomic back to September 2014 when I visited their lab. And back then, Yinzhu was the staff in Fang's lab and waiting for the become a tenure track assistant professor in the Zhejiang University. And then, so when we start this collaborator project, and so he was being trusted by his own PI, Professor Fang, and treated as our whole team member. So he involved in our project from the very beginning, know every details of our ideas and progresses, and our innovations, and particularly our problems that we encounter and the solutions. So even then after that, he went to US in February 2016, he still received the progress report from Ziyi every month almost, or every week, I forgot, until January 2017. And so our old work was repeated and modified somewhere else, without letting us know. Basically, someone took away that his lab makes hard work result of optimization and use a better instrument. So we have a OV Excel back then in our lab, and then preemptively submit the article without notifying at least the PI. And so that is the story of nanopores. And then in April 18, 2018, I actually received a letter of apology formally from my collaborator Chun Fang to explain the whole event to me, because I really mad when I heard this thing. So I don't want to show all the details. I just want to say, because our community is really cool, and then I want to say that papers and academic position are important, but not to trade off your integrity. So thank you for your attention.